After studying this module, you shall be able to learn about preparation of brain tissue for microscopic examination, learn about several strains used to highlight brain structures, learn about various anterograde and retrograde methods of labeling, learn about techniques to study living brain, learn about different types of microscopes used to study brain tissue. Neuropsychology makes use of methods derived from several branches of science and psychology. Histology has been derived from chemistry, where it involves the study of cells and tissues. In neuropsychology, great emphasis is placed on the structure and function of the brain. The structure or anatomy of the brain may be studied from 2 points of views, that is, study of gross anatomy and that of microscopic anatomy. The gross structure of the brain is visible after the skull is opened up surgically, but brain is a very intricate structure and also has several layers, that is, it is a deep structure too. It is therefore essential that methods to be employed to study it with the help of microscopes to see the intricate network of cells in the brain. This method is required to study not only the normal brain but also damaged or diseased one to ascertain relationship between brain and behavior. It is also used as a supplement to the lesion method, where the site of the lesion is to be verified after the animal has been sacrificed. The brain tissue needs to be prepared for microscopic examination. The following procedure is adopted. The first step is to do fixation of the brain tissue so that it will not change its shape. For that, it is put into formalin. Formalin is a solution of formaldehyde gas. It stops autolysis, which is the self-dissolution of the tissue, and kills microorganisms that may destroy the tissue. The second step is to put it into paraffin, which freezes the tissue and performs perfusion, that is, replaces blood with a neutral saline solution. The next step is to do sectioning or slicing with the help of a microtome, a very sharp knife of 20 microns in diameter. This section may be placed on the glass slide of the microscope, covered with a transparent liquid called the mountain medium, and is now ready for observation. Stains used to study detailed structures of the brain. The brain tissue has two basic colors, white and grey. They appear in big masses and the detailed structure of cells is not distinguishable. Several dyes have been developed for the purpose, since they are taken up differently by various parts of the cell. A few of them are described below. Methylene blue attaches to the nissl bodies, RNA and DNA in the cell bodies. Therefore, by coloring them, it indicates the presence of cell bodies. Vigert method uses dyes which attach to the myelinated fiber bundle, showing pathways in the brain. Golgi-Koch stains, also known as the reduced silver method, color membranes of axons, cell bodies as well as those of dendrites. Golgi and Kahal developed dyes which color only the glial cells, which is an important aspect of the study of brain structures. Nota-Gayak stains are used to identify the presence of dying neurons. In the histofluorescence method, brain tissue is exposed to formaldehyde gas and then seen under the ultraviolet light. It makes the adrenergic neurons shine under the microscope. Thus, after the brain tissue has been prepared for microscopic examination, it is covered with appropriate dyes and then observed for all the details. Study of the neural connection pathways of the brain. The information or the nerve impulses flow from one region to another in the brain. To understand such neural connections, some labeling methods have been developed. With the help of radioactivity, radioactively tagged isotopes are created which travel along with the nerve impulse. They leave their mark as they travel, and thus pathways of the flow of information can be charted out. Since they write their own ink, radioactivity, they are known as autoradiographical methods. Based on the direction of the flow of information, they may be classified into two types: anterograde methods and retrograde methods. Anterograde labeling methods, they indicate flow of nerve impulses in the forward direction, that is, from the dendrite to the cell body and then to the terminal buttons of the axons, while the retrograde labeling shows flow of information in the reverse direction. Deoxyglucose autoradiography. An example of anterograde labeling is the method of deoxyglucose autoradiography. In this method, radioactively tagged glucose is injected into a part of the brain. It is taken up by the cells, but it cannot be metabolized. So, it leaves its trail of radioactivity, which shows the path that it had taken up. If injected into the sensory neurons lining up the retina, it finally reaches the occipital lobe, the primary area for visual sensation. Amino acid transport autoradiography. In amino acid transport autoradiography method, radioactively charged amino acid is injected into some parts of the brain. Since cell needs amino acids to make proteins, it is taken up by the cells but not used due to radioactivity. It also leaves a trail behind as information flows from one part of the brain to the other. Immunohistochemistry, immunocytochemical method. A highly innovative technique for anterograde labeling is called immunocytochemical method. In this method, antibodies for any of the proteins of the cells are created in the laboratory. They are then attached to some appropriate dye and are injected into some part of the brain. Since antibodies attach to the relevant protein molecules only, they can indicate the presence of cell bodies which contain those proteins. The investigator first purifies a protein or a peptide in which he is interested, such as acetylcholine receptor in the monkeys. Then he may inject that protein into some other species, say rabbit, whose immune system will form antibodies to it. The investigator collects the antibodies from the rabbit's blood, chemically attaches them to the dyes and puts them into the monkey's brain. The rabbit's antibodies, carrying the dye with them, attach to acetylcholine receptors. In this manner, the locations of the acetylcholine receptors can be mapped. Retrograde labeling. Such methods are used to study the backward flow of nerve impulse or information in the brain. For example, it is well known that posterior hypothalamus contains areas for anger, rage, etc. But from where does it get stimulation for the kind of behavior may be the main question of investigation. Investigator has to go back to the origin of stimulation. Retrograde labeling is required. The following methods of retrograde labeling have been utilized. Fluorogold method involves injecting fluorogold into axon terminals. It travels back to the soma or the cell body of the same cell. Being radioactively tagged, the pathways shine and the root of information can be located. Horseradish peroxidase method. In horseradish peroxidase method, use is made of an enzyme which again makes a backward journey into the brain. Techniques to study the living brain. In order to study the relationship between brain and behavior, it is important to visualize the brain when it is actually engaged in performing certain actions or behaviors. Several techniques have been developed for the purpose. They are computerized axial tomography, CAT scan. In this procedure, the organism's head is placed in a donut-shaped ring called a tomography. On the other side, there is an X-ray detector. X-rays are made to pass through the organism's head and the amount of consequent radioactivity is measured by the detector. To increase the resolution of the brain tissue, the physician may inject a dye into the blood which will reach the brain and produce better contrast between parts of the brain. The pictures of the brain can be taken from several angles, rotating the axis by 1 degree each time, and the pictures can be printed through a computer. Exact location of any brain damage can be imagined through the use of CAT scans. The main disadvantage, however, is that it cannot be repeated too many times because of the harmful effects of radiation on brain. It is considered much more harmful for children and pregnant women. Magnetic resonance imaging, also known as MRI. It is also known as the nuclear magnetic resonance, NMR. Pictures of the brain with high resolution can be produced with this method without the use of X-rays or radiation. Use of magnetic force is made in this method. It is based on the fact that any atom with an odd atomic weight, such as hydrogen, has an inherent rotation. Generally, the direction of this rotation is fixed, but an outside magnetic field can align the axis of rotation. A brief magnetic field can make them move like tiny gyros. When the radio frequency field is suddenly turned off, the atomic nuclei releases electromagnetic energy as they relax. This change in energy is used as a measure to produce images of the brain. Any structural defects of the brain, even in some deeper layers, can be detected through this technique. Functional magnetic resonance imaging, known as fMRI. One of the limitations of MRI is that it is a slow process. Consequently, the image reveals only stationary structures. An improved apparatus called ecoplanar MRI has been devised, which is fast enough to observe the blood flowing through the brain. Rapid changes in the brain activity thus can be measured through this technique. Positron emission tomography, PET scan. It is also called tomography because the organism is made to wear the donut-shaped ring, as in the case of CAT scan. It provides a higher resolution image of brain activity. PET scans rely on the fact that the radioactive delay of certain elements emits a positron. A positron is an antimatter particle with the same mass or an electron but having an opposite charge. The first step is to inject radioactive glucose or some other chemical in the bloodstream of the organism. It has a short life. A cyclotron is needed near the PET scanner to continuously produce molecules of radioactively tagged glucose. Cyclotron is a big and expensive instrument, thus one of the limitations of PET scan is its high cost. The more active areas of the brain absorb more of the glucose which gets collected since it cannot be quickly metabolized. The collision between positron and electron emits gamma rays, which are the basis of producing brain images in this method. With the help of PET scan, location of tumors, epilepsy and other organic brain defects can be investigated. Interestingly, PET scan has also been used to study the development of brain in infants since it can tell us which areas of the brain become most active at a particular age. It was found that thalamus and brainstem started showing greater activation at a much earlier age, 5 weeks, compared to cortex and cerebellum, 3 months. Measurement of regional cerebral blood flow. This is known as regional cerebral blood flow method. When an area of the brain is more active, its effects are seen in many parameters such as blood supply, consumption of glucose and the synthesis of proteins. In this method, the investigator uses a substance that dissolves in the blood. One such widely used substance is the xenon gas, the radioactive gas. The organism is made to inhale the gas, as a result of which the gas enters his bloodstream. It reaches the brain. The areas of the brain that receive more blood will get more of radioactivity carried through the gas. The investigator placed the organism's head in a PET scanner or in a similar device. The computer then produces the brain images with higher radioactivity shown in bright red color. How does greater amount of blood reach the more active areas of the brain? It has been found that certain neurons, when active, release chemicals that dilate the nearby blood vessels. Another reason is that certain neurons, when active, release nitric oxide, which strongly dilates the nearby blood vessels. The release of potassium at the time of neuronal functions perform similar tasks. While using such methods in neuropsychology, the organisms are asked to engage in a series of tasks such as doing a heavy cognitive task or listen to music, etc. Since brain is topographically organized for various functions, the greater amount of radiation or blood flow intakes which brain areas were involved in performing those tasks. Electroencephalograph EEG. Electroencephalograph is the electric recording of the brain without opening the skull or any use of radioactivity. Electrodes are placed on the scalp of the organism and the normal wave patterns of the brain activity at specified regions of the brain are recorded and the pictures produced through a computer. Abnormalities in the EEG pattern may suggest presence of epilepsy, awake, sleep or dreaming states and excited or related states of the brain. Monopolar and bipolar recordings have been made to compare functioning of brain at two different sites or of corresponding areas in the two hemispheres. Evoked potential research has also been undertaken to study not only abnormal brain activity but also sensory processes. Microscopes. Several kinds of microscopes have been developed, for example, light microscope, electron microscope, and scanning electron microscopes. Light microscope magnifies the difference between light absorbed and light reflected by the specimen of the brain. It can show differentiation of 0.2 microns. Electron microscope is constructed to bombard the specimen with electrons. When the electrons strike the specimen, they get scattered and form a pattern. This is converted into an image of the brain by the computer. A resolution of 0.02 microns can be achieved through this microscope. Scanning electron microscopes are based on the same principles. The added advantage is that they present a three-dimensional picture of the brain tissue. Summary: histological method involves study of the structure and function of the brain cells and tissues. Brain tissues need to be prepared for microscopic examination. Stains or dyes have been developed to highlight differentiation of brain structures. Anterograde and retrograde methods are used to trace neural pathways. Techniques have been developed to study living brain, which includes CAT scan, PET scan, MRI, fMRI, and measurement of cerebral blood flow, etc.